All right, well, thanks everyone for coming and welcome to this special edition SeDNAs ECR webinar. My name is Scott and today we're going to talk about writing conference abstracts along with a few tips for presentations and posters as well. This webinar is mainly aimed at ECRs and students, but most of these things are quite useful at any career stage. So yeah, anyone of course is welcome. And the whole kind of goal of this webinar is to make the whole process of, writing and presenting feel a little less intimidating and hopefully give you some practical tools that you can use for the upcoming conference. So this webinar is being run by the conference ECR subcommittee, which includes all the lovely people you see at the bottom of the screen there. So Kristen Fernandez, myself, John Lawley, Michelle Scriver, Joe Perkins, and Josh Newton. And this is being held in the lead up to next year's conference, which is being held in Canberra from the 16th to the 19th of February. Today, we're going to focus on three main things. Firstly, improving abstract writing. Secondly, improving presentations and poster design. And third, just building confidence in scientific communication overall. So yeah, today I'm going to walk through how to write clear and concise abstracts and a little bit about structuring engaging presentations. Then we have Joe and Gracie who are going to give us their talks, which they won awards for at last year's conference. And then John is going to talk us through how to design impactful scientific posters.
Wow.
Okay, so let's start with abstracts. So this slide here is probably the most important conceptually, and it kind of explains why abstracts are important. Now, they are, essentially just a short, self-contained summary of your research, around 150 to 300 words. But, importantly for conferences, this is, the first kind of impression that people get of your work. And it's also used by conference organizers to select which talks are going to be included in the conference, also to help organize sessions, and sometimes to determine, whether you'll get an oral presentation, a lightning presentation, or a poster. So, even though it's, quite this short summary of your work, it matters quite a lot. And I think one mistake that people make is kind of treating them as, a rushed admin task that you do right before the deadline. But they're actually a really important communication tool. So this is the core structure of an abstract. So you start with what the background is, or what the context is, the broader context, then what the knowledge gap is, which links to your actual research question or your aim. Then you talk about what methods you used, what did you find, and why does this matter? And one really important thing is, this is an eDNA conference, so you don't need to use half your abstract explaining what eDNA is or, common terms like metabarcoding, for example. We all know what this is. So, use your word count for your science rather than textbook definitions. So here is an example of an abstract. This is actually mine from the last conference for a talk I did on understanding eDNA transport in hopes of enhancing box jellyfish detections. And what I kind of want you to notice with this example abstract is how, each sentence has a really clear purpose for each kind of section. So the background here is really, short, sweet, straight to the point, a single sentence. So, Chironex fleckeri poses significant risks to human health, yet they remain difficult to detect due to their transparency and patchy distribution. So everything you need to know is right there for the broader context. Now, knowledge gap, I made it super obvious. So sentence number 2, eDNA offers promising detection tool, but interpretation is limited by poor understanding of, its transport in dynamic marine environments. So that's the knowledge gap. So it's straight there, straight up. For aims and methods for my abstract, they're kind of inverse, but this kind of just shows that there's no real strict rules on how you can structure it as long as you have all this information. So for me, I first covered my methods, which was using biophysical models to incorporating decay rates, using that to simulate transport dispersal, and then led into my aims, which were to quantify transport pathways, assess environmental drivers, and compare model predictions with field observations. So the aim is really explicit. It's right there, a single sentence, and the methods are concise but informative. The results are the meaty part of an abstract. So 2 sentences spent there. This is what most people want to read, what they want to see. And so make sure, it's your big findings, and the conclusions, you want that to directly relate back to the broader context. So, bringing everything together. And, a really good abstract should be easy to follow on the first read. So you shouldn't be having to go back and reread things to try and understand what's going on. Now, some common problems. These are the most common problems that we probably see. The first is being too vague or general. A lot of abstracts can sound scientifically impressive, but after you read them, you still don't actually know what the study did. The second, no clear research questions. So people describe a topic area, but never clearly state the aim of their research. Third is missing or weak results. This is a huge one. Saying things like results will be discussed. It's usually not very compelling. Fourth, methods. Sometimes people will write abstracts where the methods are either way too detailed or there's not enough detail. And finally, too much jargon or overly complex writing. Simple writing is always stronger and gets your message across. Here is an example of an intentionally bad abstract. So this is the same abstract that I just showed you, but I've made it worse. And this kind of incorporates all the common problems which we just talked about. And, just looking at this, it looks okay. But once you read through it carefully, there's, quite little concrete information within it. And so we're going to talk through the kind of issues of this. So things that are highlighted in red, their issues are they're too vague or general. So, phrases like important to study or many things that are not fully understood, they don't really tell the reader anything specific. So they're quite general and they don't really have a place here in the abstract. A good abstract should make the reader understand exactly what was found or what you're doing. So specificity is almost always better than, being too general or trying to sound complicated. Next problem with this abstract is that there's no real clear research question or hypothesis. So if you read through this, you can work out what the topic is, but it doesn't actually really say what's being tested. So it's really important that you do include your actual aim or your specific research question quite early on. The next problem is that the methods in this abstract, the methods are really too broad and descriptive. It says, understand patterns of transport, retention, dispersal, and detection. That's probably a much more elegant way to write that. And also from this, the reader can't really work out, things like what modeling framework was used or what data were incorporated or, what the actual experimental design looked like. You kind of want enough detail that people understand the study design, but without turning the abstract into a methods section. It's a fine balance there. This is also another issue here. So weak results. So the results section here is really quite generic. It basically says, things affect other things. Like it says environmental conditions influence transport. Of course they did. The audience wants to know specifics, like what conditions, what patterns emerged, what was important, which this one doesn't include. And then conclusion is also quite generic as well. The study provides important insights. What insights? This may be useful for future research. What future research? It's just too general. So this abstract is a really good example of what not to do and can kind of show you, where those common problems usually are in abstract writing. Now, after looking at these, here we have some actual practical tips that you can use to, immediately strengthen your abstract writing. One of the biggest things, and which I've touched on, is being specific and quantitative where possible. So instead of saying something, like something had an important effect or the results were different, briefly explain how or to what extent. Your results are actually, the most important part of the abstract, so prioritize them. Give them more space, more word count to your results. Even if your project is still ongoing, you can still include preliminary findings, predictions, or just, your broader research direction. Keeping sentences simple and direct is also really important. Clear writing, again, is always stronger than complicated writing. Using active voice can help with this as well. So, saying we analyzed eDNA transport instead of eDNA transport was analyzed just makes it more clear, makes it more personable. And, finally, don't underestimate the value of revision and feedback. A fresh set of eyes from co-authors, supervisors, lab mates, peers, friends can make a huge difference. Like I will always send my abstract to my peers because I'm sure there's some little spelling mistake, or there could be a better way of explaining what I'm actually trying to do. Now, once you've written your abstract, or even better yet, before you actually start, check out the conference guidelines carefully. So, understand what the conference organizers are looking for. Stick to word limits. That's really important. Some conferences may not actually let you submit the abstract if it's over the word limit. And make sure you don't miss the deadline. Abstracts for the next eDNA conference aren't due until August 7th, but that's going to sneak up on us pretty quick based on how fast the year's going. So it's never too early to get a jump on writing your abstract. And the guidelines and instructions are actually all on the conference website, which we'll stick in the chat later on. These are the conference themes for our upcoming conference. There's quite a few of them. And so we should all pretty much fit somewhere within these. And one thing I'd really recommend doing is, reading through these carefully before, submitting or writing your abstract so you can kind of tailor it towards one of these sessions. And that's sometimes really useful because, if your work is relevant to, one of these topics, you want to be speaking in these themes, so then the right audience or the people you want to show your work to will actually see it. So yeah, before we move on into presentations, I just will ask if anyone has any questions. I don't know if I can see the chat. So if you do have any questions, just unmute and yell out.
Yeah, Scott, I've got a question. What if you want to go to the conference and you don't really have any results because you just sort of started your PhD? Um, yeah, how would you
Yeah,
write an abstract and you just want some feedback on study design and stuff?
yeah. So, I said results are important, but you can also put in predictions, or, you can just go to the conference and say, this is what I want to do to get feedback. I did that. A lot of PhD students do that. I think that's super useful because you get a more kind of whole field view from different people with different perspectives. So, in that results, just fill it up with, what you're predicting or what you're hoping to achieve. That's, yeah, really useful there. Awesome. If there's no more questions, I might move on to the next part. Sweet. All right. So now that you've written your abstract and submitted it and it's been accepted, so you are going to the conference, the next thing is, creating your presentation. And I have a few tips here for how to create engaging ones. So a strong presentation is really about good storytelling. So people remember narratives far better than, isolated data points. So, really try and come up with a nice story that flows well. Try not to overload your slides with text. Use visuals wherever possible. highlight key messages clearly. So make sure you get the points across that you really want to. Practice out loud. Practice really does make perfect. And honestly, practicing the delivery of your talk is probably more important than, making your slides 100% perfect. Once they're 90, 95%, that's probably good enough. Spend the rest of your time actually practicing. And lastly, really don't be afraid to let your personality come through. People can really connect with presenters who sound human, and who are showing, their passion and genuine interest in the research they've done. That's really, really important. And we're going to see that with the couple of examples, talks that we have coming up. And the last thing I want to say is, make sure there is some sort of consistency between the abstract you submitted and what you actually present. Because, if you turn up at a conference and you've read the conference program and you're expecting to see a talk about one thing, but the presenter talks about something completely different, that sometimes can leave a bad taste in people's mouths. So, make sure there's consistency there. So next up, we're actually going to get some great examples of some wonderful talks that were done at last year's eDNA conference. So we have Gracie and we have Joe, who both won awards for their talks. And I might let them take over and give their actual talks.
Awesome. I don't think Gracie's here yet, so I'll jump first.
Awesome. Go for it, Joe. I'll stop sharing.
Unless Gracie is here, and then she can't say it, but yeah, I'll have a quick look.
I think we're just getting her the link.
Cool. Okay. Shall I go with mine first?
Go for it, Joe. Yep.
Give us a second to share. Can everybody see my screen? Well, someone let me know. See the screen. Can you hear me clearly? Awesome. All right. All right. So yes, I'm going to talk to you today about a part of my PhD, which is looking at toxic traces. So using DNA metabarcoding to detect ciguatoxin producing dinoflagellates within 3 coastal regions of the Great Barrier Reef, Australia. First of all, I have to thank SeDNA and the conference, also FRDC as enabled the funding through a grant to be able to be here today, and also my supervisors, along with university and external funders. So first I'll tell you a little bit about what ciguatoxins and ciguatera poison is. Gambier discus are benthic toxic dinoflagellates, and they attach to macroalgae, and they can then produce potent ciguatoxins. Unlike other harmful algal bloom species that you normally see kind of planktonic and you see in the water column, you can smell them and see them from long distances, because these guys are actually benthic, attached to that benthic macroalgae and a lot harder to detect. When we get drivers for increased algal growth, such as nutrient runoff, coral bleaching, storm events or destructive species, we tend to get a proliferation in these benthic dinoflagellates. And in turn, they produce these toxins. So herbivorous species then feed on the macroalgae and these dinoflagellates, and then these toxins move up the food chain and they get more potent, they biomagnify until our big carnivorous species get it, and then we get sick from ciguatera, and Gollum worked a lot better in New Zealand. So ciguatera poisoning itself is the most common non-infectious seafood related illness in the world, up to 500,000 cases annually, and we've got quite a big case range here, 10,000 to 500,000, because there's no commercial human or fish ciguatoxin test. There's over 400 different symptoms, neurological and gastrointestinal symptoms, and there have been fatalities before, and also devastating cases of people never been able to drink beer again. Cooking and freezing doesn't kill these toxins, so they can prevail in your freezer for a long period of time. Again, just adding another element to how difficult ciguatera is. So here I just wanted to show that it's truly a global issue. So in orange are the endemic regions from a paper we published back in 2024, and in blue is where these fish are imported to. So we're having cases all over the world, but in orange here is where this most endemic, and this kind of overlaps with the species that cause ciguatera. So they're mainly found in the tropics and subtropics around the world, particularly in areas of coral reef like the GBR and the Pacific Island nations, as well as the Caribbean. We've also started to get detections and cases in temperate regions such as kind of northern Japan and New South Wales, and also in New Zealand. But don't worry, when I was in the conference in Wellington, again, this worked well. Your fish and chips are safe because it's not been in New Zealand, in Wellington. Also in Europe as well. So in terms of the distribution, at the time of this presentation, there were 8 species detected across Queensland, and they're all kind of labelled here on the right. It's important to know that G. toxicus was only thought to be the only species until about kind of the late 90s, and then we started to realise there's a lot more species in this genus than just the one. As you can see, Heron Island, we've got 7 different species detected, because that's where there's been a lot of molecular work, whereas in other regions, it's been more traditional methods such as microscopy, and they've not kind of been reevaluated since kind of these late 90s, early 2000s. So, and over the last 50 years, there's been around 2,000 cases of ciguatera reported in Australia, and although it's not common, it does account for over half of Australian seafood-related illnesses. Hervey Bay and Townsville are considered kind of ciguatoxin hotspots, meaning there's repeated cases from fish in these areas. So that brings me to the aims of my study. So I wanted to investigate the diversity of these Gambiidiscus species in 3 coastal locations of the GBR, identify any correlations between these dinoflagellates and the host macroalgae, and then have some insights into community composition of benthic toxic dinoflagellates or benthic harmful algal blooms. There's lots of different terms. So sampling regions at Townsville, as I mentioned before, Townsville is a region which had repeated cases. Gladstone, it's a new region we wanted to test. We've got some collaborators down there, and Hervey Bay, which is considered the ciguatoxin hotspot along the Queensland coast. So for each, there's 3 locations, 14 sites, and then we did 5 replicates and 1 control per site, and this gives us 84 samples in total. I'm just going to go through the methods. I won't spend a lot of time on this, as I know that plenty of us know what we're doing in terms of methods in eDNA, but it's slightly different. So we have to go down and actually get this macroalgae. So we go down with a Ziploc bag. This could be through snorkeling or grabs or diving. And then it's important to then get the dinoflagellates off the macroalgae. So kind of shake it like a cocktail, and then sieve it through a larger micron filters. And this should give us what I call a dinocolada, which is hopefully lots of dinoflagellates with our longmire and water algal mix. And that's why a lot of my PhD money went on pineapples and rum. So for the DNA extraction, use a PPLPP protocol, and then I use the 18S V8 V9 region primers. We then move on to the data process and analysis. So use the data2 pipeline to generate applicant sequence variants or ASVs, and then blasted them against Silver 18S database and also through NCBI as well. And then used a multitude of different methods to have a look at trees and phylogenetic analysis, which I'll go into in the results. And also, of course, for some stats. So just some kind of initial raw read data here. So we got 10 million raw reads, and after clean, we got 1.9 million dinoflagellate reads. I guess from here, I just wanted to highlight Gambiidiscus, which caused ciguatera. They actually had the genus that had the highest kind of read counts that we got back. Predominantly, as you can see on this, it's in Rat Island, which doesn't sound like the nicest holiday destination. But yeah, that's where kind of the majority of the reads, and in Nelly Bay and Townsville as well. So then I just wanted to have a look at all the sequences that I found, and this is a tree based on the 18S sequence, the V8, V9 region, which is 186 sequences, ISVs, that were detected in this study, along with 163 sequences from GenBank, all the different Gambiidiscus species. So we are going to quickly just go through what we detected. So 1 ASV here in this clade 6. So this is a new clade described in Japan a few years ago. And we detected an ASV for the first time outside of Japan sequence from this. It's not had a formal species identification yet. So that's why it's down as species from clade 6. So we have 15 ASVs from Gambiidiscus holmesii, which actually now is known to produce potent ciguatoxins. And then we had 13 AHVs from Gambidiscus carpenteri and 70 AHVs within this clade 2. And then we also had 87 AHVs from clade 7. And why I keep mentioning clades, I think it's important to know that sometimes we can't get down to species level, and sometimes we can. But for example, clade 3 has highly potent cyclotoxin producing species. We know these are the species that produce the highest toxins. Whereas, for example, clade 5, that we know that there are cyclotoxin activity on cell-based assays, but they're not shown to chemically produce cyclotoxins as of yet. Some species, for example, from this clade are just found in the subtropics, whereas in clade 2, we find more global and temperate regions as well. And then what we're saying, this is a potential new clade, and I'm going to go into this a little bit now. So yeah, we had a lot of AHVs that we couldn't describe. So basically put this into a haplotype network just to have a look what's going on. So as you can see, there are kind of two big clusters. At the top in green, you've got this clade 2, and at the bottom in purple, we've got what we're saying is clade 7. So I think what's to note here is that we could have a lot of different copies, RNA copies. So a lot of these dinoflagellates produce multiple of the same genes. So that might be seen in the top here with G. carpenteri. We have this dominant haplotype and then loads of singletons coming off, which are kind of a few base pairs difference. So we could be seeing actual biological change. We could have multiple different cells in the environment, or we could actually be having one cell producing multiple cells. So it's important that we think about this when we're having a look, for example, at AHV richness and other things as well, like recounts, just to make sure that we're not kind of overcompensating what we're actually seeing. With clade 7, as you can see, it's slightly different. We've got a bit more of kind of this spider web going on, and kind of another difference between the two here is that we can see in the top in clade 2 from Gladstone, whereas at the bottom we're seeing a bit more of red from Townsville. So we could be seeing a bit of a geographical split between these two groups, hence why they're slightly different on this phylogenetic tree and haplotype network. I just again wanted to highlight these this is AHV richness, and what I'm just showing here is these were found in the tropics, so we don't have much AHV richness there, but when we have a look in the tropics and subtropics borderline, we're starting to see this Rat Island, this holiday destination, and we're starting to see this high AHV richness, which again, it could be that we're at this kind of bottom of the reef into subtropical regions. So it could be that we're actually starting to see these kind of mutations more because of this geographical change, or it could be what we spoke about before with these multiple copies. Whereas in the subtropics in Hervey Bay, again, we've got quite a low AHV richness. Just to then just put this on a bit of a map, just to have a look. So we've looked at detection frequencies rather than any form of abundance, as it's quite hard to do in metabarcoding data. And what we saw is within each replicate, for example, 5 replicates, larger the circle, the more detections we're finding it. So I think the most important thing to highlight here is Gambidiscus holmesi is a potent cyclotoxin producing species. It's never been found anywhere except from Heron Island, and now we're finding it in more coastal regions, so in Hervey Bay and also in Gladstone, and this was the first time there's any Gambidiscus detected, but it shows that they're everywhere, and not necessarily producing toxins, but they are everywhere. I also had a look at some of the macroalgal hosts that they were attached to. And as you can see, there was a strong correlation on the CCA on axis 1 with Cladophora species, that's a macroalgae, and Gambidiscus holmesi. And why this is important is if we've got these dinoflagellates attached to Cladophora, for example, and we know certain herbivorous species feed on them, and we know certain carnivorous species then feed on that, we can start to understand the food chain. And then we have a mate who really loves fish and chips, especially coral trout, then we can start to see that trophic transfer and understand what areas might pose more of a risk for cyclotoxins. I finally just wanted to also say that I didn't just look at Gambidiscus. We actually had 44 different hab species that were detected across all of these regions. And this was just to show that it's better diversity calculated using the breaker. It's the similarity visualized in these PCOAs highlights that there is fine cell structure within these regions. Fine scale community structures. So I just want to come back and do some take-home messages. So this revealed the distribution and diversity of Gambidiscus across 3 locations of the GBR, including a potential new clade. It gives us insights into cyclotoxin food web pathways by correlating Gambidiscus presence with host macroalgae. and we started to have a look at fine-scale community structure. This could indicate certain locations are more prominent risks of harbouring toxin-related illnesses. Ultimately, this helps us understand the risks of ciguatera and seafood safety, and it keeps our fish and chips safe. So I just want to acknowledge my supervisors, funders, and everyone who helped with this PhD and within this part of the project. And finally, at the time, I put up 2 of my kind of recent publications, and also at the time I didn't have a job, so I was also used it as an opportunity to say that I'm looking for a job, and yeah, thankfully now I do have a job, and it turns out my boss was the one who gave me the presentation, the award for this presentation, although he doesn't remember that. Thank you, and yeah, are there any questions? But you don't need to ask me any questions.
Thank you for that, Joe. That was wonderful. I think it's quite obvious, why Joe got an award at the last conference, and the visuals in his presentation are really quite impressive, and the message is very clear.
I'm just a bit stuck. I'm trying to get those off now. I'm still all right. There we go. Stop sharing. Have I stopped sharing yet?
You're still sharing, Joe.
My laptop's just done a full. All right, how do I get off this? Apologies, 1 second. I can see it. It's just on that. There we go.
Awesome. Well, thanks, Joe. Thanks for that. Next up, we have Gracie, who's going to give us her lightning talk. This was a really impressive talk at the last conference. So, Gracie really kind of shows her passion, and yeah, I'll just let Gracie present. This is an awesome talk. So take it away, Gracie.
Awesome. Can everyone hear me okay? Sweet. I am fashionably late to this. I'm so sorry. So yeah, I'm Gracie. I'm about to finish my PhD, and I'm coming at you today from Dunedin, which is in the South Island of New Zealand. And I only had 5 minutes to give this presentation, so I actually went for a bit of the story approach, and that's what I'm gonna share with you today. So this story is called Where's Wallaby? And it's based on the beginnings of my PhD. So once upon a time, oh, it's not gonna let me, there we go. Yeah, okay. So once upon a time, there lived a wallaby called Wally. Any resemblance to a character you may have heard of is just coincidental. So Wally, unlike his cousins in Australia, had found a nice spot in New Zealand to call home, and this was because of some human-led introductions back in the late 1800s. And currently in New Zealand, Wallaby was sitting with 1 million friends distributed throughout both the North and South Islands, but he had actually travelled further than any wallaby had gone before. Just to set the scene, Wally was just thriving. He had no predators, no dingoes or anything like that. And he had a bunch of native vegetation to snack on. But as you can see, this was actually quite bad for the environment. So he was eating up all of the forest and preventing it from regenerating. And this was actually contributing to millions of dollars in losses to things like crops and pasture and the forestry industry. And so people were beginning to be motivated to eliminate him and the other wallabies. But the challenge with this was that Wally was just super, super good at hiding. He had a ton of land to hide in, and he was super mobile. He could just move around a lot. And people tried everything to find him. They tried hunters, they tried detection dogs, they tried thermal imaging with things like drones, and even helicopter surveys. But They couldn't find him. None of these worked. One day, though, people heard of this really cool tool, which we're all familiar with, which is called environmental DNA. And they realized that we don't actually have to see Wally to know where he is. He's just dropping his clues everywhere he goes with his DNA. So just to get a little technical, we designed an assay which was targeted for wallaby species, which is the Bennett's wallaby. This was in the ND2 gene region. And as well as being super specific, we also found this to be very sensitive, detecting as low as 9 copies of target DNA in a 2 microliter reaction. But the real test was still to come. Could this actually work in the field? So a bit of a toolkit was assembled to sample the terrestrial environment where Wally lived. So using water, which is quite a tried and true method, but also things like spider webs and air sampling. And this toolkit was put to test in a nearby wallaby zoo, which we do actually have for some reason. So first, filtering the drinking water of the wallabies gave consistently really positive results, and this was using qPCR. Similarly, the spiderweb collection on the outsides of the wallaby enclosures was also positive for wallaby DNA. And finally, putting up some filters on the outsides of the enclosures and leaving those overnight was also successful in detecting wallaby. And I've just got a picture here showing that we would cut out the centers of the filters and we would extract the DNA directly off of those. So this looked really promising. So now we're actually going to put this to the test in the field to actually try and find Wally. So to do this, the air samplers were given a nice little upgrade so that they could be deployed in open air settings. And the results of all of this and everything to come is in the sequel to the story because I'm limited here with the time. But in the meantime, if you are in New Zealand, not Australia, because you guys like your wallabies there, and you spot Wally or any of his mates, please report them. You can do this by visiting reportwallabies.nz. And that's everything from me. So quick shout out to my supervisors, so Neil Gemmell and also Kristen Fernandez. And also all of this was funded through the Tipu Matato National Wallaby Eradication Program. So thanks, guys. And I have my email here, so if you have questions about this project, it's progressed a lot since this work, or airborne sampling or things like that, please do reach out. Yeah.
Awesome. Thank you, Gracie.
Sweet.
So yeah, for Gracie's talk, you can see she puts, her own spin on it, adds a lot of humor to it, and makes it, she shows how passionate she is. So yeah, both of these talks are great examples of some wonderful talks about eDNA. Claire has asked if we can talk about adding humor. Humor is really important, especially in a conference setting where we're sat there for 3-4 days and it can get a bit overwhelming and tiring. So, when someone does do a talk and there's lots of humor in there, it's really refreshing. But, make sure your jokes land and, quick, witty ones are great. In Joe's talk, you would have noticed he had some little symbols where it's like, please don't take a photo. A lot of people will include that when their work's not published just yet. And so, yeah, you can include that sort of stuff as well. All right. Next up, we have John Lawley, who's going to give us a talk on creating engaging posters. So let John take it away.
Hello, everyone. Can you guys hear me well? Cool. I am looking forward to the sequel of your presentation, Gracie. That was a very good presentation. All right, let me see if I can share my screen. And I think, all right, can you guys see my screen? Yeah, cool. It's a good lineup because I think me talking about posters is going to echo a lot of what we've seen in what Scott talked about, even in the abstracts, but especially what will echo Joe and Gracie's presentation on some of the tips and some of the things around making good presentations and also making good posters. And so just a presentation I've given a few years ago, and I got kind of good feedback from colleagues that they hadn't heard about this poster design. So I hope that some or many of you can take some out of this for the upcoming conference and other conferences if you haven't used this yet. And so again, my name is John. I'll just give a quick spiel about myself for those of you that don't know me, so you knew who the hell is this guy coming and talking to me about posters. Oh, that just skipped. Oh, there we go. Sorry, I skipped a few slides. So I did a bachelor and master's back in Brazil, where I'm originally from, and moved to Australia in 2019 to do a PhD here at Griffith in the Gold Coast. I've studied jellyfish for most of my life, kind of like Scott. So did a lot of chemistry and genetics to help understand color variation in jellyfish. So that's where I developed a lot of my molecular background and kind of led a little bit later on into eDNA, which I did during my postdoc at Griffith, which I was investigating pathogen detection in agriculture and aquaculture, very different from jellyfish, but working with different systems, trying to help detect pathogens, especially using some eDNA tools as well. So that's where kind of my eDNA journey started. And I'm now out of academia working in aquatic ecology consultancy, but helping lead a lot of eDNA projects at the moment, because that was a big gap in our consultancy company, was to have people with more expertise that understood a bit more eDNA. So that's one of my main roles in the company at the moment, is working with eDNA and eDNA projects. So anyway, back to posters. And again, as I might have mentioned, this is not my idea about how to nail your conference poster. I'm just helping convey the message. But this actually came from a guy called Mike Morrison in the US that he did basically his PhD looking at user experience design and how to help use that to improve communication in science. And one of the things that happened, one of the outcomes of this PhD was this new poster design. So I'll kind of talk about, walk you through his idea and just my experience on using this poster design for the past 6 to 8 years or so in various conferences. So we're obviously familiar in academia with the concept of publish and perish, which is constantly evolving. But I think in the last 10 years or so, we're also confronted as scientists with this other kind of idea of communicate or vanish. So we not only have to publish constantly, but we also have to communicate very well, especially our research, to the public. And so again, this is not my idea of communicate or vanish. I'm just borrowing this from Marie Gardner from ThinkWell. They're also psychologists that help academics and PhD students in several ways and do research on academic performance. But this is an interesting idea because we need to think about how we communicate things to the public, but it's not just about communicating more efficiently with the public, but this leads us to think, well, how can we communicate more efficiently between ourselves as scientists, right? Because if you think about the eDNA context, we come from very different backgrounds and using eDNA for different things. So it can be quite challenging, especially for us to try to communicate the things that we are doing to each other. And so this is kind of where Mike came into this idea of improving poster design. And I'm just posting this here. The presentation is being recorded, but you can easily find his channel on YouTube to see all of his ideas and videos in more detail. I quite like his idea of the Twitter posters as well. I've used that a few times after publications to kind of make a little GIF as a summary of my publication, and you can post that on Twitter. Well, this before Twitter evolved into something else, and most of the academics evaded Twitter. I would say LinkedIn nowadays, maybe. So you can easily use these Twitter posters for posts in LinkedIn, on Facebook, and social media. All right, so going into the better poster design. So let's start thinking about what the traditional poster looks like. So this is sort of the image of the traditional poster, or the wall of text, as Mike calls that. You're basically trying to put all of your research paper or all of your ideas into that single poster. And to be honest, I think since the start of my career, I've been seeing less and less of the traditional poster design, and maybe that's the case for some of you as well. But I think as a community, we're slowly improving, especially the younger generations getting better and better at designing posters and presentations like you guys just saw. But there was a really big lag on the evolution of poster design, right? So this typical poster design with a lot of text has been around for a really, really long time. So it was kind of about time that we made some improvements to it. And so Mike gives a lot of user experience context to how he made these poster improvements. So I'll just flag some of them here for the purpose of this presentation to kind of explain how he came up with this poster design. So one of the things is this concept of desire paths. So what this image shows is that even though there's a footpath on the left, people still want to go through that little shortcut in the middle of the grass. And there might be a group of people that'll say, oh, let's put up signs or even a fence to try to block people from stepping in the grass. But from a UX perspective, you would do, well, let's put the footpath where people actually want to go through. So you're trying to make access easier for people. And of course, in our case, that means access to information. And you'll see how that relates. You can probably think about how that relates to posters. But we do have this idea that, because of our academic careers, we've been judged for a long time on how much detail we write about and how much detail we put on things. And sometimes we want to do the same with posters and presentations, put a lot of information like you would if you're writing a paper, because you think that's how people are going to judge your work when they come to the conferences. But the reality is people are pretty open-minded and pretty light when they come to conferences because they want to rejoin with their friends and have a beer. And when they come to these poster sessions, they want to learn something new. So they're actually more open-minded than judgmental, usually. And as Scott said, when you're going into a conference, you already have a lot of mental load. Like you're looking at several presentations a day, networking and trying to catch up with people. So we want to make the poster a lot easier for the viewer than maybe what a traditional poster does. And so this information foraging theory is another concept that you can think about. The way that people are looking for information and knowledge in a poster session is kind of similar to the way a caveman would look for fruit in different bushes, right? So if you think about each poster as a different little bush on a poster session, you look for these little bits of information that kind of relate to you, and you want to go and forage those bits of information. Like you can see the little caveman there go into that bush full of berries. But if that bush is really thorny, if it's really hard to get to all of those berries, you're just going to jump into the next bush, which is called that patch switching. So, oh, that's too hard. I'll just go into the next one. And you can kind of see where I'm going here with the presentation, right? So if you have a lot of information, if your poster is really thorny and not really friendly, it might discourage people actually from coming to your poster and looking at your poster and maybe even engaging with you. Another concept kind of related to that is the information scent. So the information scent is just those berries that the viewers are looking for in your poster bush. They're kind of like looking for those keywords or those images that might relate to maybe their research or their interests. And that normally comes from your title, which is where the scent is coming from in the image there in the top. So that's kind of, especially for your traditional posters, that's the main source of your information scent. But if all of this is really difficult, if your information scent is really weak, in the sense it's just coming from the title, there's a lot of blockages for people to actually come and engage with you and read your poster, because they're going to have to talk to you. They don't really know what your poster is about. They got a little bit of an idea from your title, but maybe that's not enough for the person to come and interact with you, because there's a high interaction cost. And so when you put all of this together in the context of a conference, high interaction cost, weak information sent, a lot of time pressure, because sometimes you have like 300 posters and you only have an hour to go through all of them. This can lead to a bit of a negative attitude. And I've seen this before in the past, but maybe not so much in the last few conferences that I've seen, because I think a lot of this is improving. But I've seen people in the past going like, oh, I just don't have enough energy to go to a poster session anymore if it's at the end of the day. Oh, I'm just going to go to the pub and go have a drink with my friends instead of going to the poster session. So it can kind of lead to this negative attitude, and we're missing an opportunity to convey information, to give people knowledge about things. All right, all of that to say, what can you do to improve posters, right? So you probably have an idea based on what I've been talking about. One thing that you can do is, if your main source of information sent is coming from your title, you can give a little bit more information in your title. So you might think that, oh, I'm going to make my title kind of a bit mysterious so that people get interested and they'll want to come talk to me, but I don't want to give away all the information so that people see it and just go away. Actually, you want to do the opposite. You want to give your take-home message in your title, because that already gives people all the information that they need. So perhaps in this case, instead of saying using linguistic analysis tools to study teams in ICE environments, you just say linguistic analysis tools can predict social conflict in teams. So then you have a stronger information sent already there if you just improve your title into a take-home message. So at the end of the day, if you don't like the better poster design and you say, oh, I still like my traditional poster design, maybe think about improving your title and putting it as a take-home message, because that'll make it a lot easier already for the viewer to know what your poster is about. And maybe it'll make for a stronger incentive for the person to come and talk to you if they want. Another thing is making the title or your language simpler, because whoever's viewing your poster, they're going to have to do that inside their head anyway, right? So they're going to have to kind of translate whatever complex title maybe you have into something simpler. And as I was saying, there's a lot already going on in conferences. So the easier you can make it for the viewer, the better. So if they don't have to translate it in their head, if you already translate it for them, that'll make it a lot easier. So perhaps in this example here, instead of saying bad moods distract people at work and lower their performance, you can simply say at work, mood affects attention. So that conveys your message a lot quicker to your viewer. Obviously, adding images also helps to make your poster more striking. And other things that you can do, and I think they come third here, but are really important, is just kind of adding character and fun things to your poster, because that will not just make it easier for people to understand your poster, but it conveys emotion to people, right? And it doesn't mean that you want to make people cry when they see your poster, but humor is a sort of emotion, right? So doing things like Joe did with the Smeagol, with a little Smeagol memes in there, it just conveys that humor and it brings people's attention to it because they can relate and they have some emotion towards that. So that also helps with kind of the cognitive load and understanding your message. So I think that's really important as well. Adding color, adding some sort of humor that's attached to your message. And so this is kind of the anatomy of the design that Mike came up with of this better poster. So it looks a little bit more like a billboard than your traditional poster, right? So you have really big and really simple your main findings in the middle, and a lot of contrast, because that'll drive people's attention to your poster. So you have a lot of contrast between your text in the background, in the middle there, and just your take-home message, which would be your title from in your old traditional poster. So just simply your take-home message in the middle, nothing else, you could add a graph or an image. So you'll see there's variations of this design that I'll show in a little bit. So there's a couple more suggestions here, which is you can add a presenter bar on one side, for example, that if people want to come to your poster and they want to read more about it, then they have a little bit more information in there that they can gather by themselves. Again, suggestion here to use bullet points rather than, or really summarize text rather than a lot of text on this side of the poster if you want to use it. And you can also have kind of a little clue side here for you. So if you're presenting the poster and there's like a specific graph or specific table or specific values or text that it might be hard to remember and that you want to show people that are more interested in your poster, you can kind of have that little ammo bar so you can show it to people if you want to. But at the end of the day, a lot of people don't use this ammo bar and kind of just extend this to one side. So you kind of have a bigger area to put your main findings here and maybe another image. And what you can do is add a QR code here for people that want to read your research. Again, kind of like Joe did at the end of the presentation. You add the QR code. If you want to know the full details, all the information, you can scan and save the paper to read later, etc. So I'll just show a couple more design options. So there's also, in a similar way, a portrait option. And I'll confess I've used more this design than the landscape one, just because of sometimes how the poster sessions are organized. Sometimes they don't give you a lot of width or a lot of or the panels are not that large to fit a landscape view. So sometimes the portrait view works a little bit better. But the idea is the same, is that people will look at your poster and they'll start top to bottom, left to right. And so if you have your main finding in the top, they kind of just have, if they're just passing by your poster, they kind of have 5 seconds to look at your poster, really. So that'll be the main thing that catches their attention. And if they don't decide to come to your poster, at least they know the take-home message of your research. So if you go in, there's now a better poster collection based on what, so Mike's changed a few of the designs and based on people's feedback. So there's a few different kind of design ideas based on what you prefer doing, but kind of, the idea is the same, is that you're minimizing text, adding a lot of contrast and putting your main findings first and center rather than having a lot of detail, which again, I think it's a good parallel with the presentations, right? You can see that in the presentation, both Joe and Grace didn't add a lot of information and a lot of text about their methods. It was mostly images trying to illustrate what they were doing and the results. This is obviously the striking one, right? So if you can do this figure hero poster, if you have this one image that gives you 1,000 words, that's even better. And this poster was so impactful in a conference that probably everyone has kind of seen that image in some shape or form. And it really increased the impact of the research of the person that was presenting this poster, because it was in magazines and concerts. People have, shirts made out of that pattern. So it's, again, it can really increase the impact of your research. Sometimes we think that, design doesn't really matter. The content is what matters. But for communicating to other people, to the public, to other scientists, your design really matters in how you're conveying your message. And so maybe this is what that poster would have looked like, the previous poster, if it were a traditional poster, and it probably wouldn't have had as much impact as it had, because that kind of idea and the image would have been lost in that content. So a few more examples. This is kind of already following that better poster design, that there's a take-home message kind of there at the top. Volunteers frequently looked at forehead and hair when determining a person's race. So that's already your take-home message. This is Mike's further suggestion on how he would change that poster to make it more impactful. You kind of have the hair and the forehead there on top of the poster to kind of make it a little bit fun, which is related to the message that you're trying to convey. And again, just your not your title there in the top, but your take-home message front and center. These are some examples that I've used in the past, and maybe part of it is still me hanging on to the traditional poster. So, you know, the bar is pretty low for all of you out there. I'm not saying that I'm a poster expert or I haven't won any awards for these posters, but I have won a lot of engagement and people come in saying, oh, it was a lot easier to understand or to look at your poster when I come to talk to you. So just the take-home message there in the top and some text in the middle. Part of it also is maybe my supervisors were a bit reluctant with the better poster design and they wanted me to add a bit more text. So that could help, that could happen as well. So, you know, you kind of find that middle ground in terms of adding a better design, but still adding a bit of text. So if people come and read your poster, when you're not there, they can still kind of understand a little bit more detail of what's going on. I normally like putting an image of myself in these posters, and not because I think too highly of my own image, but just because if people come and they see your poster and you're not there, they kind of know what you look like if they want to talk to you afterwards. So I like when people put their photos in posters, because I might just, you know, between presentations, I might go and have a look at the poster sessions. And if there's an image of the person there, who's the author of that poster, I can kind of chase up that person later for chat if I don't know exactly who they are by their name. So just another, maybe just another tip. This is another, I've used a landscape design before. I think that one was a little bit better. I like that one because it's just a small image summarizing and the main message in the middle. And this is another example that was in Mike's examples of posters. So if you go to the poster collection in Mike's YouTube channel, you'll see he's got now dozens of examples of posters of how people have used his design. And so it gives you some ideas on how to apply that. Again, trying to use bullet points on these sidebars and still making it more summarized rather than having a lot of text. And that's it.
Wonderful. Thank you, John. Hopefully everyone's been able to take something away from that and from our talks. I'll just share my screen again quickly. Hopefully you can see my slides. Thank you everyone for coming. We do have this QR code here, and I'm putting the link in the chat as well. We would love if you would be able to, scan this. It goes to a Google form, and we'd love to kind of get your information so we can create some sort of mailing list. So then when the next conference comes around, we can, better plan and get ideas for what you guys want to actually see at the conference, and then maybe actually hopefully create like a WhatsApp group or something where, if you find the good coffee place, you can share it with everyone, or perhaps the good bars as well. Yeah. So if you could fill out, that would be awesome. And yeah, finally, thanks for joining. Hopefully you learnt a lot. Thank you to our two speakers. Thanks to John as well for your presentation. Yeah. Cheers, everyone.
Hey John, I was just wondering, did you to get the information for the presentation, is there any links that we could share? Is that you said it's online and there's all
Yeah, so if you go to, I can share the link here in the chat.
Yeah, it'd be great, man.
Yeah, I put the QR code up there, but obviously like in the middle of the presentation. I'll just share the link to, I'll just share the link to the video of the better poster in YouTube. and that will lead you to Mike Morrison's YouTube channel, and he's got a lot of like citations and references in the comments in the videos as well. Kind of the video is like your main resource, and he in the links in the description of the video, he gives you links to all kind of the citations and research papers he based his design on, but also for all the templates and reference material. Actually, I'll share them. There's an OSF page. So all those, all the resources, so the better poster templates, etc. Share that link as well.
Awesome.
There's an OSF page.
Posts often report getting better conversations, not just more. And I've experienced this myself with my own better posters.
That's the one. Yeah, I was curious if anyone had heard of the Better Poster